Hello everyone. Today I'm going to talk about unusual benthic fauna associated with the whalefowl in Monterey Canyon, California. What is the whalefowl? When whales die and their carcasses sink to the bottom of the sea, known as whalefowl, it provides abundance of nutrients for deep-water creatures. Whalefowl along the California margin suggest that the community succession through three stages. First, the mobile scavenger stage, comprised of about 40 species, remove more than 90% of the soft tissue, usually within 4 months after arrival on the seafloor depending on the size of the carcass. Second, the enrichment opportunistic stage, comprised of about 20 species, may last as long as 2 years. Finally, the sulfuric stage is dominated by microbial mates and more than 100 invertebrate species may last for years or even decades depending on their size of the whale. Based on the numbers of whalefowl discovered during the past several decades, these habitat islands may occur relatively frequently in time and space, so we are wondering if organic enrichment due to whalefowl could play an important role in regulating animal abundance and diversity in deep regions. Canyon exploration and sample collection video surveillance were accomplished using the ROV Tiburon. It's equipped with HMI lighting, a suite of sensors to measure conductivity, temperature, depth, oxygen, and imaging sensor. Video recordings were from a high-resolution color digital camera. Digital still images the data of abundance estimates and bacterial mat coverage areas were taken from the Tiburon using two different digital cameras. Sediment push cords and bone samples and animal specimens were collected with the manipulated arm of the Tiburon. Animal identifications were accomplished through either morphological or molecular characterizations. The skeleton was at 2,891 meters depth in the axis of Monterey Canyon. It's a red star in picture. The surrounding floor of canyon was composed of fine-grained classic sediments. Trash and seagrass were observed near the fall, suggesting that this is an active sediment area. The whale was identified as the gray whale. Based on diagnostic building structure and confirmed by 99% of DNA sequence similarities of known gray whales. The length is about 9 to 10 meters. Based on weight to length communication, the biomass is about 20,000 kilograms because gray whales reach sexual maturity at 11 to 12 meters, so this specimen was assumed to be a juvenile. This animal maybe died during the round-trip gray whale migration from the full-reach Arctic, Bering, and Chukchi seas to the breeding grounds of Baja California. The remains of the whale include flesh, intestine, and baleen. The skeleton was intact, suggesting relatively recent arrival on the seafloor. Bacterial mass was developing near the skull, maybe promoted by the sulfate produced from microbial decay of lipids. The dominant animals associated with the whalefowl were two unusual polychaetes. Annelid A occupies a basal position among members of family, with only 85 to 88% AD-NASR DNA sequence similarity to other Cymbalonid genera. The 12 to 15% difference between Annelid A and other members of the group provides good support for the novelty of this species. Annelid B, the analysis of ribosomal 18 sequences revealed that Annelid B showed 86% similarity to two selected genera of the family Cybaridae. It's likely a new species or even a new genus within the family Cybaridae. Other species observed from the wellflow polychaetes such as large green tuberine amphitrachs dominate the near settlement community. It's worth noting that many of these polychaetes are thought to be a new taxa. Decapar crustaceans appear to dominate the scavenging components of the wellflow. Time 2 is 1 month after Time 1. The whale skull was still easily recognized, but ribs and other skeletal parts had collapsed to the substrate. Flesh and intestines were not as prominent as at Time 1. Associated fauna at Time 2 was very similar to Time 1. The area covered with bacterial mats had increased. Time 3 is 8 months after Time 1. Very little soft tissue remained, and many bones were degraded. Coverage by bacterial mats had greatly expanded and was heaviest in sediment near the carcass. Underlying and near sediments were black, and core samples smelled strongly of sulfide. Annelid A still appeared to be the dominant animal. Annelid B, which was abundant on previous species, was nearly absent. New animal species now observed at time points 1 and 2 include large polynoid polychaetes, 3 species of Cyparunculus, and a large nematode. Glassite crabs and large amphitrachid polychaetes dominate the surrounding sediment, which were in much lower abundance than at times 1 and 2. Discussion. Individual wellflow communities may differ greatly depending on the location, depth, and group of available colonizers. We assume that the delayed degradation of Monterey wellflow community results from its depth. Increased pressure and low temperatures may promote organic preservation. Annelid A and B at the wellflow appear to be specialistic. Exclusive use of such temporary habitats would require rapid growth, early maturity, high reproductive rates, and great dispersal capabilities. We observe high density of echinoderms on sediments near to the carcass. They are common suspension and deposit feeders in the deep sea, and may be utilized fresh organic carbon material resulting from the breakdown of whale carcass. Monterey Canyon is an active sediment transfer conduit that promotes the rapid movement of recent sediments into the deep sea. The deposition of large animal remains in this material transport conduits has the potential effect of diversity and structure of benthic communities. Conclusion. First, the soft tissue of the whale will gradually disappear. Second, the area of the bacterial mass will increase from 1 square meter to 6 to 15. Finally, two new species were discovered, annelid A and B. Annelid A was found only on bones. Annelid B was in tissues and surrounding sediments. Thanks for listening.
Thank you for your presentation. And I think there are a lot of hard terms in this slide, such as at page 9. Yeah, there is decapod crustaceans, and I know this means crabs. Yeah, right. But I think there may be audience didn't know this word, and so with a lot of hard terms, and there are less picture or enemies in your slide, so I think you can add some picture.
I should add some picture, the expression of the words to PPT, let audience better know what I want to express.
Toby.
I think your font size can be enlarged.
The font size can be enlarged.
Thanks for your presentation. And can you change the slide to the conclusion? What is these two new species?
He want to know these two species is what? This is annelid A and this is annelid B.
Thank you for your presentation. It seems like you have to present a bunch of information in only 11 minutes. So I suggest maybe you can consider to reduce some information and that is not significant. And you can slow down a little bit because for me it will be more comfortable to catch up.
I can reduce some not significant information and slow down, let her know better.
All right, thanks for your presentation. And I think overall the content of the presentation is pretty interesting. And since it's a case of a whale fall, so we can observe a lot of like phenomenons through it. But I think yes, just as what Joey has said, that the content is a bit too much, and I think you need to have more confidence. Since I have saw you like you're simply just reading your notes and your text. So yes, I hope that next time you can have more confidence and try to express what you want to say through the PPT instead of just simply just reading.
I should have more confidence, not just look at my notes.
As mentioned by Archer, actually it is a very interesting study, but wherefore in general is a very interesting topic. But okay, but I think you have not really telling the key issue or key finding of this study, which is this is the deepest whale fall ever found. All right, that's the deepest whale fall. What was the water depth? Right, this is the water depth. So that is the deepest we ever know to find a whale fall. So next time, please emphasize this. And then second is also, yeah, now the audience knows very well. So the images. You have a lot of creatures with difficult names for non-biologists. So if you want to say name a feature, please give us its images, okay, to save confusion from non-biologists. And then furthermore, the
paper you selected was actually a very nice material. However, you did not take advantage of it. So I always say, if you have a figure like this, the best way you present, can I edit it? Why can't I edit it? Oh, I see. The best way is to make it super large. Maybe this large. Put this one. This large. Okay? So that if you do that, it's the feeling that a big picture give the audience is totally different from a small photo like this. So for each one, you can use a single slide and display as big as possible to make people maybe feel disgusting or interesting. That's the power of these kind of images because they are rare. It's not something you see every day. All right. And then regarding images, because I have listened to waterfall topics several times, so I would say you probably can find images for this from the internet. So that would be more interesting than this kind of tables. Although it's nice that you summarize, I guess, from the paper, but there are better ways to present this, to use images from the internet. And then next, you talk about this material method, but I would say you should introduce the study area first because suddenly you talk about Kenyan, and I don't know how you feel. I was like, why suddenly you talk about Kenyan? So you should probably explain what was the study area, and then this was too detailed. You don't even need to say like a still images. Okay, that was too technical. You can simply add images of these ROV. That's fine. And then the text was font was too small. Avoid unnecessary space here and there, many places. Okay, and then it's good that you mark the key parts of the feature with these lines. Make sure you do it next time while enlarge your images to real large ones. Okay, and then regarding your conclusion, the conclusion was, I think you can probably remove some of the discussion here where you don't show photos. For example, because the main key issue of this paper is photos. For example, do you have photos from echinodons from the paper? If not, you can probably skip it. And then regarding conclusions, I think you can do it better next time. The second part should be results, not conclusion. You were just saying, okay, we see microbial mat here and there, but you didn't provide number in the result section. Next time, please put this into result. And next time, the better way to present a conclusion is to probably you give us a table saying, okay, in this super deep well-fall site, what is the first observation in Taiwan? And what's the second? And you have to tell us what's the time difference between time 1 and time 2. It's 1 month, right? And then what about time 3? What was the difference between the 3 times? All right? So this will make your conclusion very specific. Good. So hope to see your improvement next time. Thank you, Rebecca.